Good morning, everyone. My name is Congcong Lu. I'm from Food Agriculture and Forestry University. And my presentation topic is about genome evolution of the primary endosymbiont bacteria aphidicola in aphids. Aphids as agriculture and forestry pest feeding on plant phenotype, but it facing nutritional limitation because the phenotype lacks several essential amino acids. So nearly all aphids must rely on it in endosymbiont to provide them with essential amino acids, vitamins, and other nutrients. As the diagram shows on the right, the green is represented obligate endosymbiont Buchnera aphidicola. It's located in special bacterial site and only exists in Aphids. And it is symmetrically arranged in abdominal hemorrhacing of aphids. And the red represented the facultative endosymbiont such as Serratia, Regular, Verbachia, and others. A long-term co-evolution between aphids and the Buchnera has been proposed in 1993, but only with the 14 Buchnera strains. With the accumulation of data, there are some problems. In 2011, Huang and others proposed that maybe aphids and Buchnera do not have parallel evolutionary relationship in higher level, because different aphids in different ages will have different biological characteristics, and it will exert different selection pressure on their Buchnera. So Buchnera will have different evolutionary rate. So it is important for us to explore the evolutionary patterns of Buchnera aphidicola in different ages in a phylogenetic framework. And it will help us to understand the evolutionary process between aphids and the Buchnera. So we proposed our hypothesis include the first one. The genome features and the evolution of Buchnera are primarily determined by the taxonomical status or biological characteristics of their host aphids. The second one is, as Buchnera evolves, its genome gradually undergoes reduction to reach a balanced state, and the smaller genomes tend to lose more functional genes, which may be associated with the feeding behavior, life cycle, or social behavior of the aphids. And the third one is about facultative and co-obligate symbiote will replace some functions of Buchnera, leading to genome reduction of Buchnera. And we have collected our samples in China, and a total of 29 samples were sequenced using second-generation sequencing technology. And we have assembled the clean reads for Buchnera genome. Finally, we have obtained complete genome sequence of all Buchnera, as well as the sequence of the leucine plasmid and tryptophan plasmid from partition samples. And we have additionally downloaded 61 Buchnera genomes from NCBI database. Additionally, and a total of 19 Buchnera genomes from 14 subfamilies were included in the next analysis. The phylogenetic analysis showed that Buchnera strains from the same aphid subfamily were formed into a separate branches with high support, but Buchnera strains from different aphid subfamilies do not correspond one-to-one with their host aphids, indicating that the evolution of Buchnera genomes may be not directly associated with the taxonomical status of their host. So we can get a conclusion that aphids and the Buchnera do not exhibit a parallel evolutionary relationship at the family level. And we can say that as Buchnera evolves, its genome size from 600 to 500 kb and evenly reduced to 400 kb, and its CDS nurse and number show a gradual reduction trend. And the GC content of all Buchnera are very low, from 18.3% to 27%. The average is only 23.3%. And the reduced genomes tend to have an increased AT content or decreased GC content. The categories COG analysis show that the number of COG functional genes in Buchnera also show a decreasing trend, declining from 598 to 341 genes. And the 26 functional categories cluster revealed that differential patterns of autologous gene reduction during the evolutionary process of Buchnera. And there was no gene group into the follow function categories such as B, W, Y, and Z. These functional categories are related to structure and stability. These functional categories may be along with the aphids will control the Buchnera's structure stability. And the functional clustering analysis shows that the pathway about DNA metabolism, RNA, carbohydrate pyruvate, and TCA or energy metabolism, and the biosynthesis of amino acids. These pathways are relatively conserved, but other pathways have lost different genes. We can say that the gene loss in Buchnera genomes is not a reading process. And the mutational analysis of single copy autologous genes show that there was no significant difference in mutation among different groups, and all genes were affected by purification selections. But the average Ka to Ks of non-single copy autologous genes was only 0.58. Interestingly, there are 14 genes under positive retained more non-synonymous mutation site, and these genes are related to member-related metabolic pathways. This implies that their potential role in crucial signaling and metabolic interactions between Buchnera and the host aphids. And it may be a process of adaptive evolution of Buchnera. Our future directions include we will design different specific primers of Buchnera, and we want to see the transmission process of Buchnera. And we will dissecting bacterial-site tissues for transcriptome sequencing and to see genome expression pattern in bacterial site among different aphid lineages. That's all. Thank you very much.